Good morning, students. So, as you know that our unit reproduction is going on. So, in this unit, our third chapter is human reproduction. So, in this chapter, we'll study about the various processes involved in the human reproduction. First of all, we'll see sexual dimorphism. What do you mean by this term? Sexual dimorphism in human beings, when male and female individuals are differentiated externally, and the phenomenon is called sexual dimorphism. So human beings show sexual dimorphism. So characters which distinguish the male and female externally are known as secondary sex characters. Here's a chart is given in which you can see differences between male and female on the basis of secondary sex characters. So you can read this, is very easy. And then we are taking the next topic. So in this chapter, major reproductive events are, first of all, major reproductive event is gametogenesis. Gametogenesis means the formation of gametes, means it includes spermatogenesis, formation of sperms in males, and oogenesis, formation of ova or eggs. Next is insemination. It is a transfer of sperms by the male into genital tract of the female. And third one is fertilization. That fusion of male and female gametes to form zygote is called fertilization. Then the next is cleavage. Now, as the zygote is formed, then a rapid mitotic divisions take place in zygote, which convert the single-celled zygote into multicellular structure called the blastula or blastocyst. And next is implantation. Now, implantation means attachment of this blastula or blastocyst to the uterine wall, endometrium of the uterus. And then gastrulation. Gastrulation means it is a process by which blastocyst is changed into gastrula with three primary germ layers. Three primary germ layers we have already studied in 11th standard also: ectoderm, endoderm, and mesoderm. And these three layers are going to form various organs in our body. Next is organogenesis. Organogenesis means it's the formation of specific tissues, organs, organ system from these primary germ layers. Means ectoderm makes some organs in our body, endoderm, mesoderm, all the organs are made from these different three layers, organogenesis. Then parturition means childbirth. It involves expelling of the body from mother's womb. The uterus में से baby का बाहर expel होना, that is called parturition. So childbirth. These are the major reproductive events. Various hormones, various processes are involved in this. We'll study one by one. First of all, we'll study human male reproductive system. This is the diagram of human male reproductive system. In this, the main following parts which are present in this. First of all, we'll study of scrotum. Scrotum is a pouch of deeply pigmented skin divided into two separate sacs. So each sac contains one testis. The normal temperature of testes in the scrotum is about 2 to 2.5 degree less lower than the internal body temperature. This temperature is ideal temperature for the developing sperms. When the body is chilled, the smooth muscle contacts and brings the testes closer to the pelvic cavity. And when the temperature drops, movement towards the pelvic cavity allows the testes to absorb heat from the rest of the body so that sperm cells don't become chilled. So scrotum, which remains connected with the abdomen or pelvic cavity by inguinal canals. So next we'll study about testes. So testes, which are primary sex organs in man, during early fetal life, the testes develop in the abdominal cavity, but during the 7th month of development, they descend into the scrotum through inguinal canals. So there's a pair of testes that are suspended in the scrotum by spermatic cords. Now we'll study about the internal longitudinal section of mammalian testes. This is the most important diagram of internal median longitudinal section of mammalian testes. And first of all, we'll see that it is surrounded by three layers. First is Tunica Vaginalis. You just watch outermost layer, Tunica Vaginalis. The function is it's outer covering of the testes. Next is Tunica Albugenia. The function of Tunica Albugenia is it's a fibrous covering surrounding the testes and situated under the Tunica Vaginalis. Next is Tunica vasculosa. It's consists of network of capillaries surrounded by delicate connective tissue which lines the Tunica Albugenia. So inside the testes, there are testicular lobules. Each testis has about 250 compartments called testicular lobules. And inside the testicular lobules, there are present each testicular lobule contains one or three highly coiled seminiferous tubules, and wall of each seminiferous tubule is formed of a single layer of germinal epithelium. Next, you see that when all the these combine together, seminiferous tubules combine together, then they come out in the form of retitesti, and then retitesti joined to form epididymis, epididymis joined to form vasa differentia, and vasa differentia, and then ejaculatory ducts, and then comes out urethra. Now we'll study the detailed structure of seminiferous tubule. This diagram you're watching, wall of each seminiferous tubule is formed of a single layered germinal epithelium, and the majority of cells in the epithelium are cuboidal, which are called male germ cells, which are called spermatogonia. And at certain places, you're watching that tall Sertoli cells are present. These cells support sperm cells and provide them nutrition, especially spermatids. Sertoli cells secrete ABP, androgen binding proteins, and concentrate testosterone in the seminiferous tubules. So the cuboidal cells undergo mitosis to produce spermatogonia. Spermatogonia grow into primary spermatocytes which undergo meiosis, producing haploid cells, first secondary spermatocytes, and then spermatids. The latter convert into spermatozoa as cells. Spermatozoa means sperms. Sertoli cells provide nutrition to the developing sperms. This is again a part of seminiferous tubules in which you are watching the layer basement membrane, then germinal epithelial cells. Germinal epithelium cells is cuboidal in nature, and germinal epithelium cells which undergo mitosis to produce spermatogonia. And spermatogonia grow into primary spermatocytes which undergo meiosis. Means primary spermatocytes is undergoing meiosis. Meiosis means it's convert the into haploids. Diploid cell converts into haploid. Four haploid cells are formed. First, secondary spermatocytes, and then spermatids. Now, the spermatids convert into spermatozoa, means sperms, and Sertoli cells, the larger cells which are present. These larger cells support the sperms, and then they provide nutrition to the developing sperms, and sperms are stored till ejaculation. Now we'll study next interstitial cells and retitesti and epididymis and vasa differentia next lecture.